Welcome to the Evolutionary Biology Genetics Part 1 unit. In this unit, we're going to get into the molecular reasons behind the diversity of life that we see. So one of the things that's really challenged people over time is trying to explain the diversity of life, which is why this lecture starts with the pictures of things like the birds of paradise and all of those really beautiful sea slugs, these areas where we see not only a lot of diversity, but the diversity that we do see involves things like really dramatic colors and investing energy in making all of these structures that don't necessarily have a direct link to survival, a direct link towards making those organisms fit into their environment. So when we look a little bit at the history of genetics, one of the early perspectives was that traits were inherited, these observable structures, these observable, measurable things were inherited, but that usually with the inheritance you have blending. Of course, people's understanding of inheritance was deeply influenced by agriculture, whether that was animal agriculture or plant agriculture. Agricultural practices had a really big influence on people's understanding of genetics. So we still see a lot of this understanding of genetics today. If we just look within domesticated dogs, there's such a wide variety of different breeds. Now remember, a breed is not a species. A breed is a group of individuals with very similar genetics who should have some predictable outcomes. And the reason why I'm not saying that's always predictable is because within a breed we know there can be variation. And while you may be hoping for a specific thing, you may not necessarily get that. So within the domesticated dogs, we have a lot of very different breeds. And maybe something somebody would want to do would be to breed a pit bull. And maybe they don't like as much of the assertiveness that pit bulls can have. And so in the next generation, they may want to tame that trait down. So maybe they would take their pit bull and mate it with something like a Newfoundland. Newfoundlands are notoriously calm. They're the most mellow dogs. They were originally bred to have the traits of being calm. They would hang out with sheep and goats and hang out with the herd, protecting those animals from coyote predators. So taking something like a pit bull and breeding it with a Newfoundland, you would expect the puppies to have maybe a more mellow temperament, but Newfies are huge. They're very big, massive dogs. So maybe your puppies would also be a lot bigger than the average pit bull. So our understanding, or the historical understanding of genetics, was very much influenced by this mating of individuals to try and get predictable traits in the next generation. It was with the advent of technology that allowed us to get more of the molecular mechanisms, to get closer to causation for what we see in the next generation. So this is where we come down to our understanding of gene expression. Now, when we talk about gene expression, we are focusing in on eukaryotic celled organisms primarily. And the reason why I want to make that distinction is because if you go further, there may be some differences. So this is just sort of a general overview of how many eukaryotic cells go from the information in a DNA molecule to making a protein. Now, if you remember, we talked about in the origins of cells, we talked about this idea that you need the four big organic molecules: carbohydrates, proteins, lipids, and nucleic acids. Nucleic acids will be DNA and RNA. And this led us to the discussion of which molecule needed to come first: the DNA molecule, the RNA molecule, or the protein. And the debate or the discussion in that really comes down to our understanding of gene expression in eukaryotic cells. So we know that the DNA molecule has the four nucleotides: adenine, thymine, cytosine, and guanine. Those four nucleotide monomers, in all the myriad of different combinations, is what gives us the diversity of life on this planet. It's the information for making all of the feathers or colors or bones or eyeballs or whatever the trait or feature may be. So everything comes back to the information in that DNA molecule. But the DNA molecule itself doesn't make these things. We need to go through at least 2 processes to take those nucleotides and turn them into something like hair color or bones, or where you put your arms on your body, where you put your legs, how many fingers you have at the end of your appendages, or whatever all of the organism's parts may be. So with gene expression in eukaryotic cells, we also call this the central dogma of cellular biology. It is the 2 processes of transcription and translation. So, our first process that we want to cover is transcription. So transcription is a process of reading the gene to make messenger RNA, starting to get that information transformed so it can eventually become a trait. Now, the place that this happens in a eukaryotic cell will be the nucleus. The reason why is because the DNA molecule is stuck inside of the nucleus. So whenever we're dealing with DNA in a eukaryotic cell, we have to be inside of the nucleus. Now, there's just one structure that's going to do all of the work of reading the gene, reading the coding structure of that gene, and turning it into messenger RNA, and that's going to be a structure called RNA polymerase. So just because you've taken the DNA, read it, used that DNA to make messenger RNA, doesn't mean you've got a trait yet. The next thing that you have to do is translation. So that's our second big process of gene expression. With translation, this is going to be happening in the cytoplasm of our eukaryotic cell. And there's just one structure that's going to do all of the work. The structure that's going to do all of the work is the ribosome. It's going to grab that messenger RNA, it's going to read it and start to put together the amino acid chunks, start to make that great big long protein. So there we have it, gene expression, transcription and translation. Transcription is first taking the DNA molecule, using that to make messenger RNA, and then our messenger RNA is used to make our protein or our observable trait. Now, the next thing that we need to talk about is we need to talk about how cells can control gene expression. One of the amazing things is when we have a multicellular organism, every single cell inside of that body has all of the genes to make that organism, except for the gametes, but that's a different discussion. So when I think about my own body and I take a look at my skin cells, it's weird for me to think that these skin cells have every gene to make me, to make my bones, to make my red hair, to make the hydrochloric acid that's secreted in my stomach to help clean my food. So what I'm grateful for is that my skin cells are not reading and expressing the genes to make hydrochloric acid. That gene expression is being reserved or only being done by the cells inside of my stomach. And at the same time, I'm really, really glad that my stomach cells are not reading the genes to make hair. That would be very strange to have inside of my stomach. It would lead to a whole lot of problems. So what we see in multicellular organisms and single-celled organisms as well is that there is a lot of control of gene expression. Gene expression can happen throughout the cell's lifespan. So just because it is reading a gene today doesn't mean it's going to be reading that gene tomorrow. When you think about your own development, I hope that you appreciate the fact that not all of your cells are still doing all of their gene expression. For example, if your bone cells were still reading and expressing genes to grow you taller, you would never stop growing over the course of your lifespan, and that would be kind of weird. At some point, they stopped. They said, okay, we've reached the point that we're supposed to be, as high as we're ever going to get. Those genes got turned off. And so there you are. You're going to be this height until you get older in life, when weirdly enough, you might start to shrink a little. Well, that's a whole other discussion. So this is why we need to look a little bit at how cells are controlling gene expression, because this will help us to understand not only the diversity of life that we see right now, but why we would see diversity or see changes within individual organisms, within populations, and within the larger species category. So we're going to start our understanding of controlling gene expression by first looking at the parts of a gene. So the first thing that we define is the coding sequence. This is going to be the part of the DNA molecule that RNA polymerase reads. It's going to use that to make the messenger RNA that makes the protein. That's why it's called a coding sequence. It's going to tell the ribosome which amino acid monomers to bring in to make that protein. The other parts of our DNA will be something called the promoter. The promoter is going to be where RNA polymerase can attach. And then we're going to have our operator, or sometimes it's being called the regulatory sequence. And this is going to be the part that can act like an on or an off switch. Now there's going to be two big categories of proteins that are going to be important for controlling gene expression. The first will be activators. And our other category will be repressors. Now before we get too bogged down by details or start to freak out that this is getting kind of confusing, come back to what the words mean. Start to root yourself in what you know about a code. Right? A code usually means that it can be interpreted and made into something, promoting something. We want it to happen. Operator, regulation. Regulation, rules, stopping, controlling. Activators. Making things happen. Activation. Repressing. Stopping things from happening. Holding things back. So if you start to feel like things get a little bit complicated and you're like, no, science words, always come back to the idea that you probably know what the root of the word means. Start to work your way through that and maybe it won't seem so intimidating. Okay, so we've got a lot of different parts of our DNA, and then we've got these extra things that come in and can control things. So let's talk a little bit about how this can happen. Ultimately, if we want a gene expressed, we want RNA polymerase to read the coding sequence. In order for that to happen, our RNA polymerase is going to start at the promoter. If we don't have anything in the way, we don't have any repressors latched onto that regulatory sequence, RNA polymerase can go zoom, 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 zoom. So let me give you an analogy. Imagine that this circular saw is RNA polymerase. We know that this can zoom along, doing all kinds of work. As it zooms along, it's going to produce a lot of stuff. Okay, it's going to produce a lot of sawdust, but it's the best that I've got for an analogy right now. When this thing is just hanging around like this, it's useless. RNA polymerase is the same way. So what I need to do is I need to get this thing activated. I need activators to get my circular saw working. The way that I can do this, at least in my analogy here, is I can plug it in. Once I get the electricity flowing, it's still not going to run. All right. So, I've got my circular saw. I'm going to go plug it in. That's going to get it activated. This is my DNA sequence. Right now, it's not plugged in, no activators, no transcription is happening. If I get it activated, then it will transcribe, but as many of you know, they all have this extra safety button. So not only do I have to have it plugged into the electricity, but I also have to press on the safety button. I've got to release that, get it out of the way. Once all that happens, I can read my coding sequence. I can cut my board. So, if I have activators present, and I get rid of my repressor, my gene can be transcribed. So some gene expression is that way. It happens that way. Other times, I don't want my gene expressed. I add my safety switch, I take out the electricity, and nothing can happen. Essentially, my gene expression is turned off. And that's really what you're looking at when you start to get into your case study on the stickleback fish. With the stickleback fish, we're looking at different types of activators, how they allow RNA polymerase to read the gene expression to be turned on, and the structure to be made. We're going to be looking at how different structures latch on and stop gene expression from happening, turn that gene off. So keep that in mind when you do your stickleback fish case study. If you have any questions about that, please let me know. I think this ends up being a lot more intuitive if we don't get hung up on the terminology. There's a lot of different ways to turn genes on and turn them off. Ultimately, we're trying to get to that structure, get to that trait. A couple of other things we need to talk about. We're going to start to walk away, get a little bit bigger. One thing that we need to remember is that one strand of DNA has lots of different genes. So when we start to look at gene expression, when we start to look at traits being inherited, we have to keep in mind that if we are selecting for one trait, let's say we go back to the Pitbull Newfoundland example, if we select for one trait, like being less aggressive, that we may inadvertently get other traits as well. The reason why is because the trait that we're interested in is due to a gene on a piece of DNA, but maybe further down that piece of DNA, we've got other genes for other traits. Some of those other traits may be coat coloring or size of the skull, or anyway, we will get into that in the second half of this semester. Right now, I want to introduce you to the idea and get you used to the idea that our DNA has lots and lots and lots of genes. Not all of those genes are actually going to be directly making proteins. And that's another key concept that we need to keep in mind. DNA can have lots and lots of different functions. We're still learning about all of the other things that are included in the genome, even if they aren't direct coding sequences that are directly coding for proteins and observable traits. So that's getting us kind of back to the bigger picture again. Let's dive in to another key concept here in genetics part 1 that has to do with mutations. So when we start to look at mutations, a lot of times we think of mutations as being bad, disease, awful things happen when mutations in DNA occur. And that's simply not always the case. That's a bit of a misconception and baggage that we bring to the concept of mutations. Mutations can lead to diversity. And so when we look at humans, we know that there's a lot of diversity in eye color. Recently, scientists discovered that the blue eye color was due to a mutation in 1 or 2 individuals that then became more frequent in the population. Now, what's also interesting about blue eyes and the mutation is that this tends to be a recessive trait. But if you go to someplace like Ireland or Sweden, you're going to find that blue eyes are very frequent in the population. So just because it's a recessive trait doesn't necessarily mean that it can't be the most common everywhere. Everybody's got it. So mutations can lead to diversity. Mutations can be the adaptations that allow organisms to survive. Make sure you've got the definitions of the mutations and the examples provided memorized. We're going to keep using this throughout the semester. Our final discussion of mutation, after we get through those sort of common mutations that we see and some examples of the outcomes, will be the radiation breeding. This is where humans are artificially manipulating DNA, artificially causing mutations to occur, and then those mutations can lead to interesting outcomes, and we see this a lot in botany and horticulture, where radiation breeding is bringing us a lot of new flower colors that have never existed before. So we have to start changing our brain as far as the concept of mutations are concerned. A lot of the adaptations that we will be talking about and seeing throughout this semester are due to mutations. And this is why you covered the icefish case study. That case study shows us that sometimes having a mutation in the DNA is a very positive thing. The reason why I have you do the icefish case study first is because I find that case study is a little bit more tangible than the stickleback fish case study. So please make sure you do the icefish case study first and then get into the stickleback fish case study. I think that ordering will be easier on your brain. So once you've got diversity in your genes, what other ways do we have, or how can we pass those genes on to the next generation? Well, earlier we talked about how with cell division mitosis, one cell will make two genetically identical cells if we're talking about eukaryotic cells. So if one cell has a mutation, through mitosis, it can pass that mutation on to the next cell. If we're dealing with prokaryotic cells, the process of binary fission, one cell making two genetically identical cells, is how they can pass that mutation on to the next generation. So that's great. But we have to have more ways or other ways to get genetic diversity other than just mutations, because as I mentioned, mutations are very rare in the population or in cell division. And so if we want to increase the possibility of genetic variation, we've got two main processes. So for creating variation within a species, when we're not doing mutations, we're going to break our understanding of this into two big categories. We want to focus in on what eukaryotic cells do. And what eukaryotic cells are going to do is they're going to do reproduction. Remember that characteristic of life. Living things will reproduce. They will make genetically new combinations. Now, when eukaryotic celled organisms are going to make genetically new combinations, get some genetic variation, they're going to go through a lot of steps. The first thing that they're going to have to do is a cell division process called meiosis. Meiosis is where we take a somatic diploid cell. We've got two pieces of DNA for each chromosome position. We've got diploid cells, and we're going to split the DNA in half. We're going to decrease the amount of genetic material in the gametes. One cell will make 4 genetically non-identical gametes. They have to be genetically non-identical because the gametes, whether sperm or egg, are going to be haploid. So if we have an organism that has 60 pieces of DNA in the somatic diploid cell, at the end of meiosis, those haploid gametes will have 30. If we have an organism that has 46 individual pieces of DNA in the somatic diploid cell, at the end of meiosis, they will have 23 pieces of DNA. This is what makes the gametes not genetically identical. So you go through the specialized type of cell division to make gametes, sperm or egg. And most multicellular organisms take it one step further. They have one individual organism that only makes sperm and a separate individual organism that only makes eggs. So you go through the specialized type of cell division, you make haploid gametes. Great. That's not a genetically new combination. You've just gotten close, but the gametes are far away, you haven't made the next generation. You've now got to go through fertilization and bring the gametes together. And this is where we get into all of the amazing and really, I think, energy-intense and erratic behavior. Great big antlers and running around. And we just saw this video on the chamois in the Pyrenees where the males will not only spread urine all over their body, but they also spread sperm all over their body. I don't know if this is the perfume to attract the females or to ward off other males. Nobody really knows. But they go through this very stinky process. Chase each other all over. Lots and lots of energy. Sometimes they chase each other into exhaustion. Very, 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 very scary stuff. Just to get the gametes together. Now, because our species does this, we seem to think like this is the way all organisms should do it. Meiosis to make gametes. Lots of energy to get the gametes together. But when we take a step back and we start to look at the bigger picture, the question has to be, why would you invest all this energy? Why, as an individual, decrease your own fitness just to get the gametes together? Like, why is that so successful for creating variation in the babies, the next generation? Because you don't get to reap the reward of all of this genetic variation. You're passing that on. Maybe the kids will do better. And then we have to really step back and go, well, do they care about their kids? I mean, wait. So when we get to the chapter about sexual selection, we'll start answering some of those questions, but I have to tell you, we've got a long way to go before we can answer all of those whys. I still think that this is a very, 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 very dramatic way to go. Clearly, it does create a lot of genetic variation, but from the individual's point of view, this is a very problematic prospect. So then let's look at prokaryotic cells. What do they do? Well, if we're thinking about prokaryotic cells like bacteria or archaea, they're going to do something a lot easier. They're essentially going to use a structure called the conjunction pili. And what that conjunction pili allows them to do is to swap genes. So they can slide up to another bacterial cell. They can start to push out that conjunction pili, make a connection, and molecularly start to ask each other, hey, do you want this gene? I got all these great genes. I've experienced some life. I've made some mutations. You want them? And the other cell can say, yeah, I'll give you these genes. And they can start to swap genes back and forth. This horizontal gene transfer is very, very powerful for creating genetic variation within the species. A lot easier than meiosis and fertilization, like a lot of multicellular eukaryotic organisms do. So this really rounds out our understanding of how we create genetic variation, how we start to get some of that diversity in life, going all the way down to our molecular understanding of traits and adaptations and what we start to see and measure and observe. In part 2, we're going to get into the Mendelian genetics, that sort of modern understanding of genetics. We'll learn how to use the Punnett square tool. Hopefully this is stuff that you've reviewed before, so it comes back to you quickly. As always, if you have any questions, please, please, please make sure to email me. Drop into Zoom office hours.